Researchers optimize gene editing for SCD and beta thalassemia. Researchers at Dana-Farber, Boston Children's Cancer and Blood Disorders Center and the University of Massachusetts Medical School have developed a strategy to treat two of the most common inherited blood diseases, sickle cell disease and beta thalassemia, applying CRISPR-Cas9 gene editing to patients' own blood stem cells. Described this week in Nature Medicine and in a January report in the journal Blood, their approach overcomes prior technical challenges, editing blood stem cells more efficiently than in the past. The two studies show that the gene-edited cells generate genetically corrected red blood cells producing functional hemoglobin. We think our work defines a strategy that could lead to the cure of common hemoglobin disorders, says Daniel Bauer, MD, PhD, an attending physician with Dana-Farber, Boston Children's and a senior author on both papers. Combining gene editing with an autologous stem cell transplant could be a therapy for sickle cell disease, beta thalassemia and other blood disorders. Together, sickle cell disease and beta thalassemia affect 332,000 conceptions or births worldwide each year, according to the World Health Organization. Both diseases involve mutations in the gene for beta-globin protein. In beta thalassemia, the mutations prevent red blood cells from producing enough of the oxygen-carrying hemoglobin molecule, leading to anemia. In sickle cell disease, the mutation causes hemoglobin to change shape, distorting red blood cells into stiff, sickle shapes that block up blood vessels. More efficient editing. The Nature Medicine study used CRISPR-Cas9 technology, in particular a Cas9 protein modified by a team led by Scott Wolf, PhD at UMass Medical School, to optimize gene editing. In previous attempts to edit the genomes of human blood stem and progenitor cells, the efficiency, specificity and long-term stability of the edits once the cells engraft in the bone marrow have varied. The new technique improves the targeting and durability of the edits. Efficient editing of the blood stem cell population, ideally at rates approaching 100%, is critical to achieve a durable therapeutic effect for patients, says Wolf, a professor in the Department of Molecular, Cell and Cancer Biology at UMass Medical School. Progress toward this goal has been advancing through the contributions of multiple laboratories in the scientific community. My research team, in collaboration with the Bauer Laboratory, focused on improving the efficiency of delivery and nuclear entry of the CRISPR-Cas9 technology to achieve nearly complete therapeutic editing of the entire blood stem cell population. Bauer's team used the strategy to make a highly targeted edit. Previous work at Boston Children's had showed that inactivating a gene called BCL11A allows red blood cells to keep producing a fetal form of hemoglobin even after birth. Fetal hemoglobin doesn't sickle and can stand in for defective adult hemoglobin. More recently, Bauer found a safer target, a genetic enhancer of BCL11A that is active only in red blood cells. With our new very efficient protocol, we can edit the BCL11A enhancer in nearly all blood stem cells we collect, overcoming some of the technical challenges of editing these cells, says Bauer. In our experiments, more than 95% of copies of the enhancer sequence were changed in a way we expect would be therapeutic. The strategy enabled mice carrying blood stem cells from patients with sickle cell disease to produce red blood cells with enough fetal hemoglobin to prevent cell sickling. The team showed that the gene-edited cells, infused back into the bloodstream, engrafted in the bone marrow and produced genetically corrected red blood cells. Later, when blood stem cells were isolated from these mice and transplanted into other mice, the cells engrafted again, still carrying the therapeutic gene changes. Applied to blood stem cells from patients with beta thalassemia, the same strategy restored the normal balance of the globin chains that make up hemoglobin. The other study, published in Blood, used a similar gene editing protocol to target forms of beta thalassemia that involve splicing mutations, errors in bits of DNA near the beta-globin gene that change how the gene is read out to assemble beta-globin protein. In this study, 9 patients with beta thalassemia donated their cells, which were manipulated in a dish. For some patients, the UMass team produced a different enzyme, Cas12a, to more effectively target their mutations. The CRISPR system efficiently made edits and restored normal splicing of the beta-globin protein in blood cells from each of the patients. Setting the stage for a clinical trial. The investigators are taking steps to take their BCL11A enhancer editing strategy to the clinic. They are developing a clinical-grade, scaled-up protocol for cell product manufacturing, and performing safety studies necessary for regulatory approval from the FDA. They plan to seek funding from the National Heart, Lung and Blood Institute's Cure Sickle Cell Initiative to launch a clinical trial in patients. Dana-Farber, Boston Children's has already begun a clinical trial of gene therapy for sickle cell disease. That approach increases fetal hemoglobin production by exposing patients' blood stem cells to a lentivirus carrying instructions to knock down the BCL11A gene in red blood cell precursors. Bauer believes it's important to keep pursuing both approaches. These conditions are very common inherited diseases and occur especially often in parts of the world where resources are quite limited, he says. So we need a broad set of therapeutic options to make treatment available to as many patients as possible. Yuxuan Wu and Jing Zheng of Dana-Farber, Boston Children's are co-first authors on the Nature Medicine paper, Bauer is corresponding author. Shuxian Xu of Dana-Farber, Boston Children's is first author on the Blood paper, Bauer and Wolf are co-corresponding authors. The work was supported by the Translational Research Program at Boston Children's Hospital, the National Institutes of Health NHLBI, NHGRI, NIAID, NIGMS, NIDDK, Bluebird Bio, the Harvard Stem Cell Institute, the St. Jude Children's Research Hospital Collaborative Research Consortium, the Burroughs Wellcome Fund, the American Society of Hematology and the Doris Duke Charitable, Charles H. Hood and Cooley's Anemia Foundations. Most precise measurements of sickle cell disease building blocks could lead to new treatments. In a breakthrough study of sickle cell disease, biomedical engineers in the University of Minnesota College of Science and Engineering have revealed that the building blocks of the disease are much less efficient at organizing than previously thought. The findings open the door to new treatments, including new medicines that could be prescribed at lower doses, for the approximately 20 million people worldwide who suffer from the lifelong disease. The study, which includes the most precise measurements ever of the disease at the molecular level, is published in Science Advances, a journal of the American Association for the Advancement of Science. Even though it has been known for decades what causes sickle cell disease at the molecular level, no one has ever studied the disease at this level of detail, said David Wood, an associate professor of biomedical engineering at the University of Minnesota and a lead author of the study. What we found at the nanoscale was quite surprising. We found that the disease self-assembly process is less efficient than we thought, which means that it could be easier to develop new medicines that would be effective at lower doses and would cause fewer side effects for patients. Sickle cell disease is an inherited lifelong disorder that causes problems in the protein within red blood cells, called hemoglobin. The hemoglobin molecules carry oxygen throughout the body. With sickle cell disease, hemoglobin molecules form into fibers that act like stiff rods within the red blood cells. The formation of these fibers stiffens the red blood cells and can change the shape from disc shape to crescent, or sickle, shape. When the red blood cells stiffen, they contribute to blockages in blood vessels that slow or stop the flow of blood. When this happens, oxygen can't reach nearby tissues. The lack of oxygen can affect the entire body, causing severe pain, increasing the risk of strokes, and causing infections. The University of Minnesota researchers in this study performed the highest ever resolution measurements of single sickle hemoglobin fiber assembly in action using microscopes and cameras that can measure the molecules at the nanoscale. Their measurements show that the rates of sickle hemoglobin addition and loss have been underestimated in previous studies. The new results reveal that the sickle hemoglobin self-assembly process is very rapid and inefficient. They found that the process is 4% efficient versus 96% efficient as researchers previously thought. It's kind of like building a tower by stacking Legos, Wood said. For every 100 Legos only 4 actually stay on as part of the tower. I would have to stack another 100 Legos to get another 4 to stay on. This shows us that this is a much more inefficient process than previously thought and that it wouldn't take as much medicine to disrupt this process. Currently only 2 FDA-approved medicines are available to those with sickle cell disease. While newer alternatives to existing medications are now in development to treat the disease, including stem cell transplants and gene therapy, Wood said those treatments will probably not be available to the millions of people in the developing world who suffer from the disease. Globally, it is estimated that 300,000 infants are born annually with sickle cell disease. In the developing world, it remains a major killer of infants and children, particularly in sub-Saharan Africa and India, where an estimated 50 to 90% of infants born with sickle cell disease will die before age 5. In the United States, nearly 100,000 individuals have sickle cell disease. The Centers for Disease Control and Prevention estimates that sickle cell disease affects 1 out of every 365 Black or African American births, and 1 out of every 16,300 Hispanic American births. The U.S. median life expectancy for those with sickle cell disease is 47 years. We are hoping the new information we revealed in this study could make a big impact worldwide to develop medicines and other treatments that help millions of people, Wood said. Wood collaborated with fellow University of Minnesota Department of Biomedical Engineering Professor David Odd and postdoctoral researcher Brian Castle on this study. The research was built upon Odd's previous work studying a similar assembly of microtubules in cancer. We thought we could apply what we learned studying microtubules, which are important targets for cancer treatment, to sickle cell disease, and we were right, Odd said. This shows the power of collaborative research. My area of expertise was studying microtubule self-assembly in cancer, and Professor Wood has been studying sickle cell disease for more than a decade. By teaming up, and bringing in Dr. Castle's expertise on nanoscale imaging and analysis, we feel we have made a major breakthrough. Blood diseases cured with bone marrow transplant, doubling the low amount of total body radiation delivered to patients undergoing bone marrow transplants with donor cells that are only half-matched, increased the rate of engraftment from only about 50% to nearly 100%, according to a new study by Johns Hopkins researchers. The findings, published online Mar. 13 in the Lancet Hematology, could offer a significantly higher chance of a cure for patients with severe and deadly inherited blood disorders including sickle cell anemia and beta thalassemia. These results are really exciting as we're approaching a 90% cure rate for sickle cell and beta thalassemia, says Robert Brodsky, M.D., professor of medicine and oncology research at the Johns Hopkins University School of Medicine, director of the Division of Hematology, and a member of the Johns Hopkins Kimmel Cancer Center. Bone marrow transplants are not just for patients with a perfectly matched donor. A half match is definitely good enough. In the late 1980s, explain study leaders Javier Bolaños-Mead, M.D., Associate Professor of Oncology at the Johns Hopkins Kimmel Cancer Center, and Brodsky, researchers discovered that bone marrow transplants could potentially cure sickle cell disease, a condition with few effective treatments and one that typically kills patients in their 40s. However, this treatment has only been used sparingly since then. Until recently, it required bone marrow donors and recipients to fully match each other in a set of proteins known as human leukocyte antigens that are displayed on cells. Without a complete match, Brodsky says, the recipient's body recognizes donor cells as foreign and launches a destructive attack. Since finding a full match is difficult in this patient population, fewer than 15% have fully matched siblings free of the same genetic defect that causes sickle cell disease, and less than a quarter have full matches in unrelated registries. Johns Hopkins researchers developed a protocol, published in 2012, that allows patients to receive transplants from relatives who are only half-matched. This advance significantly expanded the pool of potential donors, Brodsky says, but the resulting transplants only engrafted to produce healthy new blood about 50% of the time. Seeking to increase the odds of engraftment for these half-matches, Bolaños-Mead and his colleagues tested a new protocol for bone marrow transplants in patients with severe sickle cell disease and beta thalassemia, two related blood disorders known as hemoglobinopathies that are caused by defects in the same beta-globin gene. They recruited 17 patients for the study, 12 with sickle cell disease and 5 with beta thalassemia, with a median age of 16. Each of these patients had a relative who could serve as a half-match for a bone marrow transplant: siblings, mothers, fathers and one aunt. As in the previous protocol, all received doses of chemotherapy and total body irradiation to knock down their immune response to the donor bone marrow before transplant. Rather than receiving 200 centigrade (CGY), the low dose of radiation delivered in the previous protocol, patients in the new study received 400 centigrays, still a relatively small amount of radiation that was well tolerated. After their transplants, all received a dose of cyclophosphamide, a drug that has proved critical to avoiding a potentially deadly condition known as graft versus host disease that is particularly prevalent with half matches. Over the next 30, 60, 180 and 360 days, and yearly after that, the researchers tested the patient's blood for chimerism, the amount of DNA present from their donors that signals that a successful engraftment has taken place. They found that all the patients, except one with sickle cell disease, had successfully engrafted, a rate significantly higher than that seen with the previous protocol. Although 5 developed graft versus host disease, the condition resolved in each of these patients. At the time of the study's publication, only 3 patients still needed to take immunosuppressive medications. All of those with successful engraftment had either extreme reduction or no symptoms of their disease. The sickle cell disease patients no longer had the pain crises that are hallmarks of their condition. Similarly, the beta thalassemia patients were no longer dependent on blood transfusions. These latest findings add to an extensive and growing body of evidence supporting the safety and effectiveness of half-matched bone marrow transplants, says Richard Jones, M.D., director of the Johns Hopkins Kimmel Cancer Center Bone Marrow Transplantation Program. We have performed more than 1,000 half-matched bone marrow transplants, and our clinical studies have proved so successful, with safety and toxicity comparable to matched transplants, that half-identical transplants must be made available to more patients as a curative option. Only mild, low-dose, or mini, therapy is needed to allow the transplant to take, making the transplant potentially safer for patients with sickle cell disease and thalassemia who are often unable to tolerate the high-dose, myeloablative therapy needed for gene therapies to take, Jones says. Other Johns Hopkins researchers who participated in this study include Kenneth Cook, Christopher Gamper, Syed Abbas Ali, Richard Ambinder, Ivan Borello, Ephraim Fuchs, Douglas Gladstone, Christian Gok, Carol Ann Huff, Leo Luznik, Lode Swinnon, Heather Simons, Stephanie Terazakis, and Nina Wagner-Johnston. Earlier emergence of malaria in Africa. Malaria, which claims hundreds of thousands of lives each year, mainly children and especially in Africa, is one of the leading causes of death by an infectious agent, the Plasmodium falciparum parasite. In research on malaria, the genetic mutation that causes sickle cell anemia, also known as drepanocytosis, a chronic disease that is often fatal in children under 5, caught the attention of the scientific community very early on because it also provides protection against malaria. After carrying out extensive research into the beta-S mutation by performing full sequencing of the HBB gene together with a large-scale genomic study on 479 individuals from 13 populations from sub-Saharan Africa, scientists from the Institut Pasteur and the CNRS were able to reveal that malaria emerged in Africa at least 20,000 years ago, and not at the same time as the adoption of agriculture 4,000 to 5,000 years ago. The findings will be published in the American Journal of Human Genetics on February 28, 2019. Individuals carrying the beta-S mutation in the HBB gene who do not develop sickle cell anemia, healthy carriers, demonstrate increased resistance to malaria infection. This evolutionary paradox, first revealed in the early 1950s, a mutation that is by definition harmful but promotes the survival of some individuals, means that beta-S can be seen both as an emblematic example of natural selection in humans and above all as an ideal marker for malaria research, since the date of emergence of beta-S corresponds with the minimum date for the emergence of malaria. Research carried out in recent decades suggests that the date of emergence of beta-S, and therefore also malaria, coincides with the dates on which agriculture is known to have been adopted as the main means of livelihood in Central Africa around 4,000 to 5,000 years ago. The scientific community had long accepted the existence of a causal link between the emergence of agriculture and the spread of malaria in Africa. But nothing was known about the history of malaria in African populations that did not adopt agriculture. Drawing on new genetic data obtained by scientists from the Human Evolutionary Genetics Unit at the Institut Pasteur, a study carried out by Institut Pasteur and CNRS scientists Guillaume Laval and Louis Quintana Mercy, in close collaboration with the Max Planck Institute in Leipzig, Germany, and the IRD, has cast doubt on the role of agriculture in the emergence of malaria in Africa. The results of this collaborative scientific research, based on a novel formalization of the specific natural selection method generally accepted in the case of beta S, show that this mutation emerged around 20,000 years ago. These new findings therefore indicate that malaria was rife well before the adoption of agriculture, contradicting widely held interpretations. The research also shows that the beta S mutation emerged more recently, approximately 4,000 years ago, in hunter-gatherer populations. Changes in the equatorial forest during this period, most likely because of an episode of climate change and/or a period of increased deforestation owing to the emergence of agriculture, are thought to have facilitated the spread of malaria among pygmy populations. We show that the beta S mutation, which provides resistance to malaria, may have been spread by agricultural populations who came into contact with these populations of hunter-gatherers during the Bantu migration, when farming communities crossed the equatorial forest and set out on the major migratory routes to the eastern and southern regions of sub-Saharan Africa, comments Guillaume Laval, lead author of the paper. These results shed new light on a little-known chapter in the history of malaria and demonstrate the beneficial effects of admixture for some aspects of public health, such as the spread of mutations conferring resistance to various pathogens among human populations, adds Louis Quintana Mercy, joint blast author of the paper.